Okay, today we're going to talk about development and plasticity of the brain. So at just 2 weeks gestation, when an embryo is 2 weeks old, the nervous system begins to form. And what we see in the beginning of this is the dorsal surface thickens, and then long, thin lips rise, curl, and merge together. And we get this neural tube surrounding a fluid-filled cavity. And you can see in D, so as we're looking at this neural tube, that fluid-filled cavity is in the center there, and that's going to become the central canal and then the ventricles of the brain. And you can see at the top there, pointing to the future brain. So a great deal of this neural tube is going to become the spinal cord. And as the tube sinks under the surface of the skin, that forward end is going to be so that part that was pointing to before, future brain, that's going to differentiate into the hindbrain, midbrain, and forebrain. And I find this set of pictures rather helpful in what's happening at about 3 weeks out, that we see that fuse and we start to see the brain differentiate from the spinal cord, and he gives us the cross sections of how this is coming together and where that central canal is. You can see the central canal on the bottom. So we're going to walk through a number of processes that that neurons go through as they are growing and developing, or being created really, and developing. We're going to start with proliferation, so the creation of new neurons. Talk about migration. The neurons have to get to the part of the brain that they are where they're going to function, and so they need to move there. And then when they get there, they need to differentiate. Some of this is happening, depends on the neuron, a little bit maybe before migration, where we get the dendrites and axons, but typically they migrate to where they're going to perform their function. So they go to where the visual cortex is, and then they differentiate into the kind of neuron that they are. And we also see synaptogenesis. So the neuron is created and it starts creating its synapses with other neurons, and myelination. And while some of this we do see a bit throughout our lives, but this is mostly discussing what's happening prenatally as the nervous system is developing. But again, some of this, we have some proliferation, let's just say in the prefrontal cortex. I'll take that example, even in adolescence. And so we're going to see this synaptogenesis, and then we have myelination. It's a long and slow process, and it can, again, we see some of this up until early adulthood, really. But I'm going to walk through each of these processes. So first, talk about proliferation, or the production of new cells. Many of our neurons are produced by the time we are 28 weeks gestation. And in fact, if we have a premature birth that occurs before that time, it inhibits some of the growth of the brain. We have proliferation a bit really throughout our lives, but this vast amount of creation of neurons and glia, that is happening very early on. And what's happening is it's the cells lining those ventricles that are dividing. So if you go back in your mind to the neural tube, it had that the center was fluid filled and was going to become the central canal and the ventricles. And really it was just this tube around that, right? So we have these cells that are lining those ventricles. They're dividing. They are stem cells. Some of them are going to stay where they are and continue to divide, and others are going to become primitive neurons and glia that then are going to migrate to other locations, to where they need to be. Okay, so after the neurons are produced, they need to move to the location where they're going to perform their function. Some of them are faster, some of them are slower in their migration. A few of the slowest don't reach their destination until adulthood. You can see in the picture at the bottom right what's being shown there. So that very bottom part, the kind of almost darker orange, is the ventricular zone, if you can't read that. So that area that's near the ventricles. And as we're early in this process, in this proliferation and movement, we see the neurons that are going to be close by are the ones that are getting to where they go. They are following these radial glia. So the radial glia are the ones that guide migration. The radial glia are coming out from the ventricular zone, kind of like spikes of a wheel, and the neurons are traveling up. And you can see that as I go to layers 3, 4, 5, 6, as we get to the far right of the cortex, that those neurons have to travel through those lower layers in order to get to reach their destination. And we have chemicals known as immunoglobulins and chemokines that are guiding that neural migration. And we have many different kinds of these chemicals, which really reflect the complexity of the brain. So differentiation is next, and it's really after migration to a large extent. When neurons are produced at first, they look just like other cells, and they're going to migrate to where they are going to perform whatever function they're going to perform, and then they're going to differentiate, forming their axons and dendrites. And it's said rather strongly because to some extent, so they can form those axons ahead of time, as some of them have these axons in tow. So they develop their axons when they are at a place where they're going to need to communicate to these neurons, to these targets, and then they continue to grow and kind of just leave that what's going to be the terminal buttons behind and bring that axon in tow. And others get to their destination, and then they have to grow their axon out towards the targets that they're going to communicate with. Neurons reach their destinations, and then the dendrites begin to form. Okay, we have synaptogenesis. So once we have those axons and dendrites and they start to meet up with their targets and communicate with each other, we see the formation of new synapses. This begins long before birth, and it continues throughout our lifetime. So we're going to see a few times this bit of proliferation of neurons and some synaptogenesis, creation of new synapses. We're also going to see times when there is synaptic pruning, as we create more neurons and synapses than we need, and then we go back and prune what is not being used, or those weaker synapses. This slows in older people, so I'll talk a bit about how this, especially in certain areas of the brain, continues throughout our lifetime, but it does slow as we get older, as does the formation of new dendritic branches. And finally, we have myelination. It starts about 24 weeks gestation, but we see a great deal of myelination for quite a while. And again, throughout as we're creating new synapses, we can see myelination for a long time throughout our lives, but a great deal of it is done in those final weeks of pregnancy. And again, the myelination is, let me not skip this. Remember, we have those myelin sheaths, those insulating fatty sheaths that are allowing for greater efficiency of communicating between neurons. And it moves in a rostral direction. So this starts in the spinal cord and then up through the hindbrain and then through the midbrain and then through the forebrain. And again, it's gradual. It continues for decades. So just an overview of fetal brain development. It begins in the embryonic stage between weeks 2 and 3 of gestation. By 4 weeks, we have differentiated the forebrain from the midbrain from the hindbrain. By 11 weeks, we see a differentiation between the cerebral hemispheres. The cerebellum and the brainstem also become apparent. And by birth, we have a brain that is about 350 grams, about 100 billion neurons, and it's almost 25% of the weight of the adult brain. And so a lot of our proliferation is done. We will see these times of proliferation. The rest of the weight that we're going to gain in our brain is not so much of the new neurons, because we're also going to lose neurons to apoptosis, but this continuing development of the synaptogenesis and the myelin sheaths as well. And we do have new neurons later in life. We have stem cells in the nose creating those olfactory receptor neurons. So as we are going to talk about our different sensory receptors, these are the sensory receptors that are there for smell, or the sense of the olfactory sense. Many species have these stem cells in the olfactory bulb as well. We're not going to see that in humans. And again, we'll talk about this a little bit really soon, as we're about to get into sensation and perception. That first area where the olfactory receptors synapse. And then we also have new neurons in the hippocampus. And I'm going to talk a little bit here about the hippocampus, and I'm going to stress this a couple times. I'm hoping I'm saying this in a way that is interpreted correctly. So newly formed neurons in the hippocampus go through a stage where they're highly changeable. They are going to need to be integrated into new circuits that are helping to represent memories. And when I say so, I think it's your author that says that represent new memories. For me, my understanding of the hippocampus is it is helping us to consolidate those memories. It is involved with bringing those memories back up. But those memories are represented or stored in the temporal lobes. So really this is the consolidation and formation of new memories that we want to be able to bring back up into our conscious recollection at this moment. At times of new learning, we see more of these new neurons survive. And I'm going to talk about kind of the care of the hippocampus, even though, as I'm about to talk about the size of the hippocampus and what it's correlated with, remember that these are correlations, that we see smaller hippocampi in older people. We see smaller hippocampi in people who have Alzheimer's. We see smaller hippocampi in people who are depressed. We can, if we, people who lose their sense of smell completely from some accident, it's pretty common that they become depressed, and we see smaller hippocampi in those people as well. So I just want to stress this, how important new learning is, okay, because that neurogenesis is happening. We can see more neurogenesis when we are doing our aerobic exercise, as we're getting this blood flow to the brain and allowing for that neurogenesis. But we not only want the neurogenesis, we want those new neurons to survive, because we want large hippocampi. In my opinion, if you want to keep your memory and be able to remember new things in your explicit memory, you really want to be able to figure out how do I develop my hippocampus, or my hippocampi as the plural. And we are creating new neurons. And again, all of this slows as we get into older adulthood. The next section is called pathfinding by axons, and this is a really good question of how axons find the correct targets. So they have to travel great distances across the brain to find the correct neurons or targets and to form those correct connections. And the question is, how do they find their way? Paul Weiss in 1924 was examining this question when he transplanted an extra limb onto a salamander. So salamanders and other amphibians, unlike mammals, they can take a new limb and it will start to just move in synchrony with the limb that's next to it. And how Weiss interpreted this was that, if I'm remembering this correctly, that those neurons were sending, like many neurons were sending signals, and that it was just that those muscles were recognizing the appropriate neurons. But that's not really what happened. He dismissed this idea that each axon was actually finding the correct muscle. But we're going to find out from his student, Roger Sperry, that that is exactly what happened. Each axon found the correct muscle. So in 1943, Roger Sperry published research where he had cut the optic nerve of a newt, so the nerve that's going back from the eye to the brain. And it grew back and connected with the tectum appropriately. And if you remember from before, we're going to get into this again, that the tectum is where our superior colliculus is, which is controlling our eye movements, taking in a lot of information so that we can move our eyes appropriately. The tectum in the newt and in amphibians is their main visual area. It's not a very interesting visual world, in my opinion, but that's what they get. They get the tectum. He then cut the nerve and he rotated the eye by 180 degrees. And I always have to do this with my hands. So flipping the eye basically upside down. What had been dorsal, the dorsal part of the retina, was now the ventral part of the retina, and vice versa. What was the ventral part of the retina is now the dorsal part of the retina. And what had been the dorsal part of the retina, those axons grew back and connected with their original targets. Okay, so it wasn't that the world stayed the way it was because the dorsal part of the retina grew back to the dorsal part of the tectum. It's flipped now upside down. So I'm realizing that's a little bit confusing because of how light works, and that it is giving us on the retina this flipped upside down image, and I just said everything's flipped. But so we've learned to interpret the world. What's coming from the dorsal retina is what's at the bottom right of our world. What's coming to the ventral retina at the lower part is what's at the higher part of the top of the visual world. And so now for these animals, and we've learned to interpret this. I mean, even though everything's flipped throughout our brain representation, we know that up is up and down is down. But for these animals, this retina has been flipped upside down. And so now what appears to be down to them, okay, is actually what's up. And so if their food is in the world down below them, they're going to reach for it. They're going to go up. If their food is over to the right, they're going to move to the left. Everything's been flipped around. So they're finding their appropriate targets, and they are doing this via chemical gradients. So that's how axons really find their way. There are some chemicals that are attracting those axons, and they have other chemicals that are repulsing those axons so that they find their way to their appropriate targets. Okay, I was explaining some of that with the previous picture, and I'd forgotten that this picture's on here, and it's really helpful. So I'm going to do just the dorsal again, but you can realize that this is left, right, anterior, posterior, all of that is flipped. So if you look at the top picture, my dorsal retina, or salamander's dorsal retina, is communicating to the ventral part of the tectum. They have severed the optic nerve and flipped this upside down so that what was dorsal is at the bottom and what was ventral was at the top. And they have a nervous system where they can create new axons in the central nervous system, unlike mammals. So again, this is partly just finding the right animal to work with. And so what happens when they regrow that optic nerve, those axons, you can see now the old dorsal, which is now at the bottom, is still communicating to the ventral tectum, thus spinning or flipping the visual world for this animal. And axons don't achieve perfect accuracy. Those chemicals that are helping with the pathfinding get them to the area they need to be in. But the question now is, how does the appropriate afferent axon connect to the appropriate target? And the answer is that we start out with more neurons and synapses than are necessary, and we see this elimination of those weaker synapses. So the postsynaptic cell is going to strengthen some synapses and eliminate others. And this is sometimes referred to as neural Darwinism, as it is the strongest links that survive, right? And this is done via neurotrophins. So if I'm communicating to the postsynaptic cell more strongly, then I'm going to receive more neurotrophins that make it more likely that those axons and those synapses, those survive. They allow for the survival and activity of neurons, and I can't remember. I do remember it was in the Freeberg textbook that she discusses one of the things it looks like we see in people who are autistic is more neurotrophins when they are younger, and so we're not seeing as much of the apoptosis, or cell death, this kind of neural Darwinism that we see in a, quote, normal brain. And it was Rita Levi-Montalcini who told us about a lot of this kind of apoptosis and neurotrophins, and how cells, some of them need to die off, and then how other ones survive. And if you read a little bit about her in the textbook here, it's very interesting that she was working in Europe as the Germans were destroying much of Europe at the time and killing lots of people. So focusing on her work, but she found, okay, working in the sympathetic nervous system, that the sympathetic nervous system forms far more neurons than it needs. When one of its neurons forms a synapse with a muscle, that muscle delivers, in this case, a protein, the neurotrophin nerve growth factor, or NGF. We will come back to that a couple of times throughout the semester, which promotes the survival and growth of the axons. And then neurons that don't make contact with the appropriate postsynaptic cell, or the ones that are arriving later, so they're just not as strong synapses, they are going to die in a process called apoptosis. And again, as suggested by, I can't remember the authors who discovered the relationship between neurotrophins and autism, but this apoptosis is really important. We need to get rid of the neurons that are not working, because we are creating more neurons than we need. So all of the areas when they are developing in the nervous system are forming far more neurons than they need, and that apoptosis and synaptic pruning, all of that loss in a particular brain area is really indicating maturation. And so I think we do a really small bit on adolescents and older people as part of this development of the brain. So for example, teenagers are going to lose cells in parts of their prefrontal cortex, and this occurs up till about age 25, which is why I do that thing at the beginning of my classes sometimes of, this is the time for developing your prefrontal cortex. You want the appropriate connections to survive so that you are able to do things like regulate your impulses, regulate your emotions, consider other people, think about your future and your goals instead of this kind of immediate gratification. All of that is still developing up to around the age of 25. And again, part of what's really important to remember is that their neurotrophins are necessary for the survival of neurons and essential for the growth of axons and dendrites to form new synapses and learning. And so I don't think it is nerve growth factor, which is one of our neurotrophins that we see, especially as the brain is developing. I think it's the brain-derived neurotrophic factor that I come back to, again, when I talk about the hippocampus, that this is allowing for those neurons to survive in the hippocampus. We don't just want neurogenesis. We don't want just proliferation. We want the neurons to be used, and the ones that are useful, we want them to survive.